Okay, good. Yeah, let's start then. Welcome everyone to our Cambridge Cheminformatics informatics meeting today in February. I will give a brief overview of those meetings in the beginning. So what are we trying to do? The aim is to bring cheminformaticians, but in the widest sense, can be modelers, bioinformatics, all of that. Let's call it cheminformatics. So where we analyze life science data, let's say, together scientifically as well as personally. We had those meetings for quite a while now, nearly 10 years, I think. Started with physical meetings in Cambridge. So it was usually 30 or 40 people in those meetings. And now it's usually 200 or so registrations from all over the world. So it's usually 50% attending. So it's usually 100 people or so participating. And usually I mention the countries from west to east, so from Japan to Brazil, but let's go north to south, or at least to the south. So especially welcome to participants from South Africa and Zimbabwe on this occasion. So it's a much wider reach, of course, we have with the meetings right now, which I'm very happy about. So welcome everyone. As for future dates, please write them down. So the next meeting will be on the 1st of June and the 2nd of November in this year. So these are the Zoom meetings. And we're also planning to have in-person meetings in April and September, depending on the situation in Cambridge. So Chris Swain, for example, agreed to host those meetings in person. So we will organize details in the near future. Please let me know if you would like to present at any of those meetings, whether on Zoom or the in-person meetings. Very happy to schedule that. Everyone is welcome to present. You can see the details of this event and future events at c-inf.net. Also, I'm sending around the Cambridge Cheminformatics newsletter, so it's about once per month with jobs and events and so on. And you can see the archived versions here at drugdiscovery.net. You can also follow me on Twitter if you use that platform in case you would like to be informed about future events. The main meeting will be recorded and put online where agreed with speakers. So that will be put were allowed by the speakers and their companies on our Cheminformatics YouTube channel. You can also go to c-inf.net in order to find that location. As for questions, please type your questions in the chat window, also along the presentation, and afterwards we will go through them orally, and speakers would ideally then follow up by chat for the remaining questions, which we cannot answer due to time reasons. And if you have a question, I would like to ask you then afterwards also to phrase it orally where possible in order to have some more personal interaction and to allow for follow-up questions and so on, to have a bit of a discussion. So if you have a microphone and could use it at that time, that would be great. If you have questions, I will then go through the questions afterwards and alert you to that. Yeah, it is important, of course, we have some talks, but there's also the social element. So afterwards, we will have the worldwide Cheminformatics social meeting, basically. And so put your beer or other drink into the fridge. Afterwards, people will go into random breakout rooms, those who can make it. And so you can meet 5, 6, 7 other people from other countries, meet your old friends again, make new friends, and so on. So stay online, please, after the last presentation as well. Because it's worldwide Cheminformatics partners to meet old friends again and to make new ones. And if you wish to present at the next meetings or have information to circulate in this Cheminformatics newsletter, please let me know. It's about 300 people or so on that mailing list right now. So there's a certain reach with the focus on Europe, but also in the US and so on. So let me know if you have anything to circulate to others in those meetings. Very happy to include that. There's one other event we organized. So that's the Digital Sciences for Drug Discovery series. So that will take place in 2 weeks, or in 2 weeks and 1 day, actually. On the 17th of February, again, it will be an event on Zoom, will be free, open to all. And you can register at this website here, DigiDrug, so Digital Sciences for Drug Discovery.net. There will be 2 presentations on this occasion. The first one will be an introduction to explainable AI, artificial intelligence for small molecules. That's Floriano Montanari from Bayer who will present. And we will have Günther Klambauer from Linz University presenting on deep learning methods for chemical reactivity. So it's a free event on Zoom, open to all. So go to digidrug.net if you would like to attend that event as well. But now let's come back to this meeting, the Cambridge Cheminformatics meeting. So we have 3 speakers today, very different and diverse areas as well. And that's what I'm trying to do as well. It's not only, well, cheminformatics, that's where we came from, but it's basically drug discovery, right? There are many topics left and right as well. And the first topic will be on automation. So we will have Charly Coulon from Invite presenting. Afterwards, we will move on to predict small molecule binding to protein structures and molecular condensates. So that's Kadi, Liis, Saar from the University of Cambridge, as well as Transition Bio, who will speak. And finally, we will have Marianna Trapotsi who did a PhD with AstraZeneca and the University of Cambridge, who will present on using cell painting readouts, so cell morphology readouts, to enable phenotypic profiling and safety prediction of PROTACs. And stay on online afterwards, please, as well. Okay, and that was the introduction. And with that, I would like to hand over to our first speaker, Charly Coulon, who will present on from integrated laboratories to orchestrated laboratories, opportunities and options for incremental automation. Please, Charly.
Okay, first I share my screen. So now it should be visible and
Looks
you
good.
should, perfect. Okay, let me arrange the window in a way. Okay, yeah, thanks for the opportunity to present our topic here today and giving us the place to, or the stage to present it to you. That's Tom and myself. My name is Charly Coulon. I started in automation 24 years ago after my PhD in artificial intelligence. And now for the last 6 to 7 years, I'm with Invite. It's the best job of my life, I have to say. And in this case, I'm accelerating the utilization of modern robotics within the industry. And the second best job has Tom, but he will introduce himself in a few minutes. Talking about from integrated laboratories to orchestrated laboratories, I would like to introduce the story first before Tom introduces Invite. If I think of the best current laboratory, in the best case, it's the way that you get all information from your lab equipment and from your automated lines into an electronic lab notebook or laboratory information management system. And in the same way, if the human is doing something, if he's transporting materials, samples, or operating manual equipment, then you get also this data into your lab system. And this is, we call integrated laboratory. And from my point of view, or our point of view, this is are the best in class laboratories in today's world. What we now think is to go beyond, so enable the lab assistant who's currently orchestrating the lab, who's currently, as I said, transporting material, executing, connecting functions which could not be automated yet, that we want to enable the lab assistant to operate the lab also during nighttime and at the weekends. So during 70% of the time where classical labs are not operated, we want to enable the lab assistant to operate his lab by operating automation. And this automation we want to provide to him will allow him to orchestrate workflows and will also provide to him additional automation to automate manual processes, like, for example, dosing, and connect the equipment and operate the equipment. And all this we want to do. We saw already implementations like this. We already did implementations like this, but in the past it was all very specific, very heterogeneous, very complex, both in the meaning of how to orchestrate or to define the workflow for the human, also in the meaning of the complexity of the technical solution generated. And our target is to simplify this to the extent we can simplify it and demonstrate it with minimized technical complexity. Before I now go a little bit more into the details, I would like to hand over to Tom to introduce himself and invite to you.
Thank you, Charly. So I'm Tom Thomas, kind of like I'm a chemist by education, a PhD chemist, and one of the tasks I have is here to lead a group at Invite. And on the next slide, I want to already start with giving you a short overview, okay, what Invite is. So INVITE is a public-private partnership between Bayer and 2 universities in Germany, the Technical University in Dortmund and the Heinrich-Heine University in Düsseldorf. We're already 10 years old, started with modular facilities. Right now we're moving on to other topics like orchestrated laboratories. So we are in essence a research center for innovative production technologies. And we also lead an innovation center, the Drug Delivery Innovation Center, which is an open consortium between partners from pharmaceutical industry and equipment manufacturers and academia, of course, all focusing on formulation development projects and activities. So our ambition is to bridge the gap between research and your application. So, you know, kind of like a lot of things, there are questions at the one hand side in industry where we don't have answers to, where some research should be done. On the other hand, we also have results kind of like reached in universities and academia, kind of like waiting for the implementation. And we want to bridge that gap and see, okay, how can we do that? And we do that always in collaboration. So we do always with the network of partners from academia and industry, depending on the topic, we pick the right partners to address it. And yeah, we try to do it either bilateral or multilateral or with public funding projects, kind of like where we want to see that we really start realizing the potential of technology innovations in an industry. We do that with a team of approximately 30 FTEs, 10 experienced experts, and many 20 PhD students, master students, people supporting us and doing research at the university. We do focus on 3 areas. Buckets here, kind of like we do smart automation, formulation technology, and innovation. So, and there we want to, in all these areas, we want to accelerate utilization of these technologies. Most of the time, not by building it completely ourselves, but by exploring them and by demonstrating them. So we do to the point where we demonstrate it and then hand it over to the relevant functions and the relevant organizations. For smart automations, we have the robotics lab, a digital showcase center where we do modular automation. And here you have then the lab orchestration platform is also part of the smart automation team. Formulation technology is a drug development, the drug delivery innovation center, and also formulation and process lab. And under innovation, we drive the topic sustainability, open innovation more broadly, and modular facilities, kind of like the things that we don't start new projects in the modular facility area anymore, but we still have some projects that we need to finish. So these are the 3 things we're doing. And if you're curious about what's happening there, we do have a open conference. The link there, go to our website, invite-research.com. And there you find the agenda and the registrations link. There we talk about the work we're doing in drug delivery together with a lot of partners. There will be very interesting presentations coming there. Also smart automation, kind of like laboratories is one part of it, a smaller part, it's only one section, but I put a red frame around it. And also a lot about collaboration. So if you're interested also kind of like, hey, how can a collaboration boost research and help to realize things faster? You will find it, I think you will get some interesting discussions and input over there. So that about getting to know us more. But we also have a very cool facility, our technical center, where we really can build these demonstrators on industrial scale. And here you see a photograph. We have all in-house. So we do have a robotics area. We do have our digital showcase center where we have kind of like miniature plants where we test modular automation technologies. And we have a laboratory where we can do to OEL4 substances where we can do some tests also with active ingredients. So we have everything in place to really test and build up the facility, build up proof of concepts and demonstrators. Now the thing is, why are we focused on lab orchestration? Why is that a topic for us? I talked already about the modular automation that we have. So we have a lot, already more than 10 years experience in modular facilities, modular automation. And if you look at the orchestration that is needed between different lab equipments, that's in essence similar, not the same, but very similar. So we have that now how that we apply to exactly the orchestration of the workflow. On the other hand side, we do have our robotics group, which is very cool in automating then some manual tasks that right now cannot be easily automated. And if you put these two together and then teams there together, you can get a lot of progress and you can realize a lot of demonstrators and find out, okay, what are the issues, where do we need to work on? And then in combination with our academic partners, finding answers faster. So that is what we're doing there. Just to give a little bit of a glimpse, what is then that modular automation? I don't know in this area if you're into it, but that's perhaps more for the electrical engineering people. There we really try to have state-of-the-art testing of state-of-the-art technology. So we have with modular type package, we do kind of like some demonstrators, find out how it works, how to do it, kind of like so. We can integrate the new package in within 10 minutes because of that. So it's quite amazing. We also do about data engineering with Namu open architecture, how you get data from certain sensors and so forth and so forth. Kind of like the wireless heart. I think perhaps a little bit too much detail here, but you see we do from connectivity to kind of like plug and play. We have that expertise and we have a lot of projects and demonstrators over there. So that is about the showcase center that we have. And I think right now it's back to Charly, who takes over and tells them, okay, if we have the orchestration of the workflows and add that with add in the additional automation, what happens then?
Yeah, thank you, Tom. And just as one add-on, which will be not in focus for today, but which is of course necessary if you want to take yourself out of the loop in the laboratory. So it's not only about your manual activities, but it's of course also about your natural intelligence. So if you take this out, you have to replace it with some artificial intelligence. So and this we do by automating the design of experiments or to optimize the design of experiments. And even though this is not part of our next phase for the demonstrator, this is something which definitely is part of the overall concept and to get the lab operated 24/7 step by step. Now coming back to the task we wanted to show to you. So it's about the orchestration and about the additional automation. And regarding the orchestration, life can be very complex, but life can be also very simple. So I didn't show to you any of the implemented network scenarios, how to integrate all the data systems with dozens of connections lines and dozens of small boxes to be explained. Mostly in these days, also somewhere a Raspberry Pi to fill in the last gap. We are convinced that we can execute the demonstrator or implement the demonstrator just by using off-the-shelf lab execution suite in a kind of way that the lab assistant can easily define the process, describe the process he wants to automate in a human readable way, but this is also machine readable in order to automate it, and that we extend, and this is non-standard today, that we extend the interfaces of such a lab execution suite. There's a sound coming from somewhere.
Yeah, could you please mute yourself if you're not speaking? I will do that from my end now as well. Yeah, thanks.
Okay. So some non-standard thing is that such a suite can connect to Zila 2 components and components integrated by UPC UA at the same time. But we think this is necessary. These are even though these are competing standards, we are convinced that there will not be the one or the other, but you always need both depending on the equipment or the automation you want to integrate. And this we will demonstrate in a plug-and-play manner. Tom shared with you that this is the target setting for us now for the last years in production, and we will use the same experiences and similar technologies to do this also in the laboratory. Finally, you can, the final target is to make it as simple as integrating a printer, an office printer, to your Windows PC or to your Macintosh PC. It might look different, but in any case, it's easy integrated in by plug and play, and this is exactly the target we envision. Now coming to the robotic steps, automation steps, which has to filled in to close the gap. So one typical step which is executed by a human is flexible dosing of substances. It's not the case that there's no automation for dosing, but those of you which know the dosing systems, they know that they are not capable of handling all the different substances, handling all the bottles, all the containers to dose in, and they can hardly dose different categories of amounts. What I mean by this is if you have a dosing system from any company and this is capable of dosing 50 milligram, it will hardly be capable of dosing 50 gram. But a human can do so. A human can dose with a simple spatula. He can dose at a scale. He can dose 50 milligram, and he can also dose 50 milligram. And we just copied this behavior to a robotics arm. We did a demonstrator to apply machine learning to this to enable the robotic system to learn by itself to dose different substances. We'll find a video and further information on our website. And this year we will work on transferring the results of the learning from one substance to the other. So if there's a new substance, that the robotic systems, based on the substance features of the substance, takes the most closest point of the parameters to start the learning and then transfer the learning results to become capable also to dose the new substance in an autonomous way. Regarding the transport of material samples and also the operation of equipment, the vision is that the same equipment which is operated by the human during daytime is operated during nighttime by a robot. And we demonstrated this in 2021. We just took a mobile robot with a 6-axis robotics arm from a supplier. The supplier also delivered the modules or the software for transportation and operation of the dedicated equipment. And then we integrated this in existing infrastructure with all these dozens of connections and bubbles and so on I just mentioned two slides ago. And all this, I'm very sad I cannot present it today. It will be presented in 8 weeks on a conference about pharmaceutical mobile robotics. But currently the slides are still under approval, so I have to point you to to the next session. But this will also part of our demonstrator will be that we extend this concept in a way that you don't need these dozens of connections and bubbles, but it's clear, straight ahead, an integration of the mobile robot, the additional automation like the dosing systems, and also the operation of the equipment, and in case we have existing automated lines, also integrating them into the overall concept. So the summary from our point of view is that there's a huge potential to enable a lab assistant to operate his own lab automatically during nighttime, during the weekend, finally 24/7, to decide by himself where to start the automation, what to automate, where he wants to get support from automated systems. We are convinced that all the necessary technologies is available, and our target setting is to simplify the overall technical concept within our current and next projects. And if you have any questions about this, of course, we are more than happy to answer them. And if not, it would be great if you can share your view to it, your feedback to it, if you see it the same way, that there's huge potential for this kind of automation or for such a concept. If you think we might think too complicated, or did we miss anything? So any feedback you have for us, we are happy to take it and to discuss it with you. Thanks for your attention. And yeah, I'm curious about your feedback. Thank you.
Great, thanks a lot, Charlie and Tom. Any questions from the audience? One problem, of course, is in practice, if you have interfaces, right, you have competing technologies, right? So how much, and if you have, for example, I mean, AstraZeneca is the iLab, right? I mean, nearly all big pharma companies, I guess, work into that direction, right? I mean, how many competing technologies do you have? What do, especially the interfaces, yeah? So what does the landscape look like? Is there a problem to harmonize, or how would you judge that right now? Is there chaos, or what does that landscape look like?
At the first look, it looks a little bit like chaos if you look at the implemented landscape. So the laboratories most interested in what we do are not those which have no automation like this, but which have a lot of automation and which are just more or less dying in maintaining the complex architecture they created. So there is really this interface jungle, or these dozens of interfaces. I mentioned Raspberry Pi, and this is not a joke. Yeah, there are really Raspberry Pi implementations to fill in some gaps where you find nothing off the shelf. So there is a huge heterogeneous landscape. But on the other hand, meanwhile, on the equipment side, so down to the field in production, we would say in production laboratory, down to the equipments, or directly to the equipments, there are really two main standards, and they are not really comparable. They are not on the same level, but UPC-UA and SEALA 2, you find nobody who would doubt that they will stay in future, and that it's necessary to connect to one or the other. Surprisingly, no one developed a lab execution layer to connect both, but there we see the solution down in the direction of the equipment, down also towards the mobile robots, down towards further automation. In the other direction, it's really, as you said, a situation which is absolutely specific for each company. So even within a pharmaceutical or within a chemical company, you might have different electronic lab notebooks and different laboratory information management systems. And this is exactly the reason why we propose to put the workflow management not inside the electronic lab notebook or inside the LIMS system, because from our point of view, on the one hand side, it's a high variety there. On the other hand side, these systems are normally not capable of handling the information needed to operate equipment fully autonomously. But if you are in the position of a chemical or pharmaceutical company, then it would really relieve your situation if you go downwards from these electronic lab notebook or from the laboratory information management system, always to the same lab execution system, and then down to the equipment from there. This is the idea behind the concept we are following. We think it's the most simple direction, but that's exactly the reason why we are here. We share this, and the better is the enemy of the good in German. I don't know if it's transferable to English, this saying, yeah. But of course, our future will tell which is the best solution.
Thanks, Charlie. Yeah, there was one question from Abi, whether the recording will be shared. Yeah, if you go to c-inf.net, the recordings will be put there as agreed with the speakers. Yeah, and there's one question from Alex Papatanasov. Alex, would you like to elaborate on your question? Yeah, otherwise I can read that as well in case you don't have a mic available. Should we focus on standards of applications? And not quite sure if it's clear which applications are meant here, and that's where maybe clarification could be useful. But Charlie, that is the question asked by Alex. Should we focus on standards of applications?
Definitely. It makes very little sense for companies having chemical or pharmaceutical business to develop their applications by themselves. And I mean, dosing substance is really nothing which occurs only at one company or one corporation. And in the same way, we are looking for standards here. We at the ISPE, the International Society of Pharmaceutical Engineers, we founded a interest group in the DACH region, so in the German-speaking region, with more than 15 pharmaceutical companies to identify those applications we all need in future. Because developing them by themselves, as probably Alex was the thinking behind this question, and the intention, is the worst case. You pay all the development, you pay all the specification, and so on. It's better if you have, of course, if there's a solution you can buy. And the best example, not for laboratories, I would even think it's about the dosing but if there are any other proposals, please go ahead. My favorite application for laboratories would be dosing and HPLC operation, but anyhow, feeding. And in production, I just want to share because I think it's interesting. It's cleaning the floor in a pharmaceutical plant. I mean, obviously everybody needs it, and obviously it's currently done by humans in the night between Saturday and Sunday. And why should anybody develop this by himself? Just currently developments are ongoing, and in future we can buy such applications off the shelf, and that's exactly where it has to go.
And it's clearly pre-competitive in that sense as well. That's right. Okay, no, very good. Thanks a lot. And one of the reasons to invite you is also there are a number of participants from pharma companies, for example. So if there's any interest, please get in touch with Charly and Tom. Thanks a lot for your presentation.
Thank you. Thanks for the opportunity. We will stay in the line. Okay.
Please do.
Thank you. Bye.
Excellent.